Welcome everyone to the very first recorded episode of Lab Rat Chat. We thought we'd mix things up a little bit on this one and give you a video option to watch during these times rather than just the straight audio, portion of the podcast that we usually do. So, it's not super exciting. Here's my water heater. There's exposed insulation. There's a concrete wall behind me. So, but it's something a little different. And so for those of you that don't know, this podcast is all about informing the public on the importance of animal research and how it is 100% necessary in order to continue to make medical advancements for both humans and for our pets. This podcast is supported by the Americans for Medical Progress through the Michael D. Hare Fellowship, which is awarded annually to support projects just like this one that educate and inform the public about the critical role of animal research in furthering the medical progress. So the fellowship honors the late Dr. Michael Hare, a renowned board-certified laboratory animal veterinarian who dedicated his career to scientific and medical advancements and who is deeply committed to animal welfare and advocacy. And in this 7th episode of Lab Rat Chat, I got to talk with Dr. Mark Slifka, a researcher from the Oregon Health and Sciences University, about his research involving developing antibody tests to help detect COVID-19 in people. And so Dr. Slifka and his team are out there working tirelessly around the clock in their laboratories on developing these tests that will hopefully be very accurate and be able to give us a better picture of the situation in our country when it comes to the COVID-19 disease. So without further ado and without butchering it any more than I already have, I'll let Dr. Slifka explain further, and we talk about all sorts of things in this episode, from tetanus to Ebola to diphtheria, so all sorts of things that you'll want to hear. So just sit back, relax, and enjoy this 7th episode of Lab Rat Chat. All right, welcome everyone to episode number 7 of Lab Rat Chat. It's been a little while now since our last episode, and as we all know, a whole lot has changed in our world between then and now. So one change you all will notice on this podcast is that until we can return back to normal, getting back to work, school, or whatever it is that might be normal for you, we'll be without Danielle as she's trying to conquer working from home and operating a daycare center basically out of her home while she's also trying to take care of her son. But don't worry, she'll be back. For now, you just get to enjoy the pleasing sound of my own voice for now. So getting to today's episode, I of course want to thank everyone out there working to help combat this pandemic and to keep our country operational, from truck drivers to grocery store workers, of course doctors, nurses, including veterinarians out there, and really just so many people that we can't go through the whole list. But one occupation that I routinely do not hear mentioned, although we hear about what they're doing all the time, are the people doing the research necessary to move us past this. There's a lot of talk about the research, but the people doing this, from the scientists, physicians, veterinarians, animal husbandry staff and care staff, research techs, research support, administrative staff, just everybody involved that are working to ensure that the research can go on, because the research must go on, deserve a huge shout out and recognition for everything that they're doing. And so with that, today's guest is a molecular microbiologist. He's a vaccine researcher. He does all sorts of types of research and is working on some pretty exciting stuff that I'm going to let him explain so that I don't butcher it trying to do it myself. So without further ado, Dr. Mark Slifka is our guest today from Oregon Health and Science University. So thank you for joining us today, Dr. Slifka, and for taking time away from your busy research schedule and your busy days to tell us about what you have going on out there in your lab in Oregon. So if you could, just tell our listeners a little bit about your background and what in your journey to kind of get you to where you are today.
Well, thanks, Jeff. I appreciate you having me on your show here. And I can begin by telling you a little bit about myself. So I got my PhD in microbiology and immunology at the UCLA School of Medicine. I did my postdoc at the Scripps Research Institute. And then I'm now a professor here at Oregon Health and Science University, and I've been here for 20 years now. My lab studies, if we want to put it under one umbrella, my lab studies immunological memory. We look at the immune responses after different types of viral infections or after vaccination. And we've been following a lot of different, in some cases, exotic viruses. We've studied smallpox, monkeypox. We looked at dengue, yellow fever. We're doing studies on Zika, and even chikungunya virus. So we look at a broad range of different viruses and the vaccines that either are made for them or vaccines that need to be made for them. One of our first studies was actually with smallpox vaccination. Shortly after 9/11, in the anthrax scare, there was concern that immunity after smallpox vaccination was only short-lived, and that immunity could only be maintained for maybe up to 3 years, and then after that was lost. That was a big concern because it meant that the entire U.S. population would be at risk if smallpox were going to be used as a biological weapon. We drew blood samples from over 500 subjects here in the state of Oregon, and looked at their immune responses after smallpox vaccination. And we were the first study to demonstrate that immunity can be maintained for up to 75 years after a single vaccination in smallpox. And so that was really remarkable. We published that back in 2003. It showed that you could maintain immunity for up to 3/4 of a century after a single vaccination.
That's great news when it comes to smallpox. That's for sure.
Yeah, and then that got us onto other similar pox virus stories. We looked at the monkeypox outbreak that actually happened in 2003. So monkeypox is the closest living relative to smallpox. And when we went to the epicenter in Wisconsin and drew blood samples from people there, we were able to identify people who had pre-existing immunity from their smallpox vaccination, and that protected them against the monkeypox. And so this is this cross-protective immunity, where we identified 3 people that had previously been unidentified during the outbreak. And by developing new diagnostic techniques, we were able to confirm cases that were previously only called probable. And so we were able to really dive into that monkeypox outbreak. And now we're very interested, of course, in COVID-19, because this is it's a huge issue. And now we want to use some of the techniques that we used in those pox virus studies to now learn more about coronaviruses.
Yeah, and there's definitely a lot to learn. It sounds like a lot of your research kind of depends on current outbreaks that are going on in the world a little bit, which is pretty exciting. I guess you're always kind of doing what's relevant at the current time in the world, and working and finding ways to help prevent, treat, diagnose diseases that are affecting the world. So that's awesome.
Yeah.
So if you could just a little bit, just kind of tell us a little bit about what you all are doing with the coronavirus, what you already know about it. It is called the SARS-CoV-2 virus, if I'm correct. And you don't see that a whole lot in the media, but it kind of makes me think that, is it similar to the original SARS that we saw back in 2003? And are we able to use information that we had studied from the original SARS virus to apply it to this one? If you just give some information about that, and then what you guys are doing specifically in your lab to help combat this or treat it or diagnose it.
Sure. So SARS, the disease SARS is from SARS, the coronavirus. COVID-19 is the name of the virus, as you mentioned, is SARS-CoV-2. And that can be a little confusing, because as soon as people hear the first part of that, SARS, they think of the original SARS virus. But they are very close related coronaviruses. They're about 85% homologous. And so we can learn a lot about COVID-19 by looking at how we responded to SARS. And so these are both coronaviruses. They're both spread by the respiratory route. They have a lot of similarities in terms of basically their biochemical structure. They're very, very similar viruses. The requirements needed to sanitize surfaces are the same between these two viruses. Even the ability to spread, their transmission rates, they have a thing called their reproductive number, which is how many people get infected from each case, are similar between these two viruses. And the good news is we were able to control SARS and clear it from the human population in just 4 months. So that was very much a success story. Now, COVID-19 has exploded and traveled around the world now. And so we have a much bigger problem now with COVID-19 than we did with SARS, because it expanded so quickly. A lot of it under the radar. But we should be able to use a lot of the same techniques that we used for SARS to battle against COVID-19. And some of those steps include direct testing for the virus itself, this PCR testing, as well as these antibody assays.
And that's what you guys are doing, right? There's the antibody assays we're trying to develop.
Yeah, and that's what we're working on now are these antibody assays. So the story is this. When you're trying to do diagnostics, there's two approaches. You either try to detect the virus or you try to detect the immune response to the virus. So during the early stages of an infection, when you have symptoms, that's the best time to detect the virus, okay?
All right.
Now, after you've cleared the infection, you typically mount an antibody response, which helps you resolve that infection. And so then at the later stages of this infection, it's much harder to find the virus because you're clearing it.
Yeah.
But now it's much easier to detect the immune response to the virus, and we do that with these antibody-based assays.
Okay. And so when you develop one of these antibody-based assays, you could be looking for people down the road that may have been infected. You could test them. They could have cleared the virus, but they still have the antibody test, and it could show that they had exposure to the virus. Correct?
Exactly. Because that's one of the main questions is, we're as we're continuing to do more testing, we're identifying people who are scoring positive for a current infection with COVID-19. But the big question is how many people went undiagnosed? How many people had flu-like symptoms over the last 2 months and yet didn't have a chance to be tested at the time that they had the virus? Now that they've cleared the infections, we can go back and look at them with the antibody assay and try and determine what proportion of the population has immunity.
Which will be very important, I think, in moving forward, with everything and opening everything back up, is kind of knowing what the herd immunity level, I guess, is, if you will, with our population. And so, but one thing is, I see on the media, just on the news or reading articles, there's lots of different tests out there that seem to be looking for these antibodies for testing people. Is one test better than the other? Is there a reason why better tests still need to be developed, or are some more specific or more sensitive? Or I guess what's the point of having so many different tests, or is it just that we just don't have enough information and everyone's racing to kind of get the most accurate test that we could possibly get to get the best picture of this disease and how it's affecting us?
Yeah. So you really touched on an important point. There are, I think I lost count there's over 70 different antibody test kits that are available that you can order to perform COVID-19 testing. But they range in terms of how good they are and how reproducible the results are. And there's a couple examples of how and why this is important. There's two, when it comes to diagnostics, there's two main components people look at: sensitivity and specificity. All right, so sensitivity means how well can you detect that particular disease or that particular immune response to that pathogen. And so you want a kit or a test that's very sensitive. If possible, you'd like it to identify everyone who had that infection.
Yeah.
That's not always the case, but if we had to have a gold standard, we like to detect at least 90% who had that disease.
Okay.
The flip side of it then is what's called specificity, and that means how specific is your assay, your test for that particular disease. And this is where it's been really tricky with these antibody tests, because COVID-19 is not the only coronavirus in town. So people have been exposed to seasonal coronaviruses. Nearly all of us have been infected with these. These are called cold-like symptoms. And we all have antibodies to these other seasonal, mainly benign coronaviruses. The problem is the reagents that they use to make these tests, those can cross-react with your antibodies to these seasonal coronaviruses, and then your specificity goes down. So an example of this is, let's say you had 90% specificity. Well, 90% is an A, right?
Right.
That's a good thing.
Yeah.
But if your seroconversion rate is actually quite low for your new disease, that can be a problem. I'll walk you through an example of this. So here on the West Coast, there's been 2 studies. They're still in preprint, so we don't have a final published version of them yet. but both in California have looked at antibody responses of local populations, and they're finding seroconversion, which means you've been infected with the virus and now have immunity to it. They found seroconversion rates of somewhere between 1.5 to maybe 6%. So that tells us a couple things right off the bat. It indicates that herd immunity is actually quite low here on the West Coast. Again, these will need to be repeated by other studies. But the part that'll be interesting is looking on the East Coast. If we take places like New York, where we have very high frequency of people scoring positive for the virus-specific test, we're probably going to have much higher seroconversion rates when they start going through with the antibody tests over on that side of the country.
Well, that's very interesting, and definitely information we're going to need to know to continue moving forward. How do you even go about starting to develop a test like this? Do you have to use an animal model? Do you need samples from known infected patients to be able to test it? Or are you working with the virus specifically in your lab and putting that into in vitro or in vivo type experiments to develop a serological test or the antibody test that we're talking about?
Yeah, there's several approaches that are used to develop these different tests. For a lot of the antibody-based tests, you look for parts of the virus that are unique to that virus. Okay. So if we were studying Ebola, we don't have any Ebola-like viruses running around the U.S. So we have several parts of the virus that can be used for that type of antibody test. Now, with the coronaviruses and the fact that we have these seasonal coronaviruses that we've been exposed to, the majority of the virus can be cross-reactive. Our immune response can be cross-reactive between these different viruses. So what we've had to do now is focus on very specific regions of very specific parts of the virus. So, for instance, you've heard about this spike protein that's on the surface of the virus. Well, there's a unique region on the spike protein that is very specific for COVID-19, where it's completely different or nearly completely different from the seasonal coronaviruses. So by taking that very specific region, we can use that as our target or our bait for these antibody tests and use that for detecting the immune responses that very specifically for COVID-19.
And then as far as, like I know for drug approval, you have to go through the whole FDA approval process and using an animal model or 2 animal models in some cases. For these tests, I know there's been some, I think, restrictions lifted in a way, or fast tracks, if you will, for this type of thing because it's so important and so critical to not have to delay the whole year. What's your timeline for all of this, and then just getting through, from the bench through regulations and then out into the public for hopefully use by the masses?
Yeah. So the FDA is giving emergency use authorization to some of these companies that produce these tests, some of the best tests. And so that's not complete FDA approval, but it's getting close. And so there's several groups that are trying to get at least that emergency use authorization. And that will help move them forward into being used on a broad scale. Now, for most of these diagnostics, they're specific to the human antibody response. So it's mainly human subject data that's required and human subject samples. But animals really play a huge role in vaccine development and also for answering some of the basic biology questions about this virus. I'll give you an example, though.
Okay.
So one of the questions that come up is there's been reports that COVID-19, the SARS-CoV-2 virus that causes COVID-19 is mutating. And it's true. And geneticists have been able to follow these small mutations and they've been able to map where a virus came from. So if a virus came directly from China to, let's say, Seattle, it'll have a unique genome sequence with a certain a number of mutations. But then as it goes through a population and then travels to another population, it'll accumulate other small mutations. And so this has been fantastic because the geneticists can look at that and say, oh, this is where this virus traveled from. And sometimes they can even determine how long it's been in a certain area by this mutation rate.
Yeah, that's pretty cool to be able to follow it around and see exactly where it's been, basically, too, if you will.
Exactly. But here's the deal. People have heard that the virus is mutating, and they're like, oh no, well, what if we can't protect ourselves against this with a vaccine? What if it mutates to a point where the vaccine won't be able to protect us anymore?
Kind of like the influenza virus, how, just mutates, and every year we're getting a new vaccine for that to hopefully help protect us against the mutated version, if you will. How it's able to do that so rapidly.
Exactly. So people are wondering, well, will this mutate as quickly as the flu does? And the answer is no.
That's reassuring.
It is. A new study, again, these are all preprint publications, which means they're available online, but they haven't undergone true peer review. But a study just came out recently where they vaccinated rhesus macaques and then challenged them with the SARS-CoV-2 virus, and they were protected against it. So that shows that you can make a vaccine against this virus. So that's very promising. But in addition to that, they took serum antibodies from those vaccinated animals, and they tested them against over 10 different strains of SARS-CoV-2. So these were viruses isolated from China or Italy or 2 or 3 other countries in Europe. And the good news is that one vaccine was able to show activity, what they call neutralizing activity, against all those strains, regardless of where they came from. And so that's also really good news, because it shows that even though the virus will undergo little tiny mutations that the geneticists can follow, it's not mutating in the locations that our immune system needs to attack in order to make a good vaccine and mount a long-term immune response. So that's an example of where the animals have been able to teach us something that we wouldn't be able to do otherwise.
Right. And which is all really good news, because there's lots of questions about, if this coronavirus might be seasonal, and is this going to be the new normal, or we're just going to go on, lockdown come every early spring, late fall. And so if there is, long-lasting protection in a way, and if you get it or you have a vaccine, then you're protected from it, that's reassuring to the future, for sure, when it comes to not having to do this again, because we all want to avoid that.
Yeah. And this goes back to one of the other reasons that we're doing these, trying to do these, they call seroepidemiology studies, or these antibody tests at the population level. If you have a good test that's very, very sensitive and very, very specific, it'd be nice to be able to tell people on the front lines, like healthcare workers, if they're immune or not, so that they'd have an understanding about their risk when they're going in and dealing with COVID-19 patients. But also we want to learn that at the population level, because if we had a high enough herd immunity, then that would actually reduce the transmission of the virus. And so one of the questions that comes up is, how much herd immunity do you need? And I can give you an example of that.
Yeah, that'd be great. I mean, I've heard all sorts of different numbers, from 30 to 60 or higher percent. So it seems to me what I've read is all over the board. So that'd be great.
Yeah. So what it does, it depends on your virus. So a virus like measles is very, very contagious, far more contagious than COVID-19, far more contagious than flu. It's got what they call this reproductive number, which is the number of people that the first case might infect.
Okay.
And so for flu, it's around 2. One person will infect 2 others. Okay. Measles, the number is like 14 or 15.
Wow.
One person will infect 14 or 15 others.
Yeah, that's a ton.
It's a ton.
Wow.
So when the epidemiologists have looked at this, in order to get protection against measles, herd immunity against measles, you have to have immunity, herd immunity of over 90%, because it's so contagious.
Okay.
The good news is that with these coronaviruses, either SARS or SARS-CoV-2, they have a reproductive number of around 2 to 3, so much more similar to flu. Okay. And the story is this: if you get that reproductive number below 1, that means that each person that's infected infects less than one new person, then your disease disappears over time.
Perfect. Yeah,
And
that's what we're aiming for then, right?
that's what we're aiming for. So if you had a reproductive number of 2 and you had herd immunity of 50%, that 2 would drop to 1. And if you got just below 1, then it goes away. And so it's good news that the COVID-19 virus is not as contagious as measles. That's really good news.
Yeah.
And so if, for instance, if we got herd immunity in the range of 50 to 60%, then there's a good chance that the herd immunity itself would reduce that reproductive number to less than 1, and the virus would burn itself out.
And I mean, I guess it's too early to even say what a timeline for that would be. I mean, at this point, right? It's not like that's over the next, 1 to 2 years, maybe, to where you'd see it burn itself out and be gone, kind of, in a way.
And that's where these studies will be so important, because again, if we look here on the West Coast, the shelter-in-place initiatives and social distancing occurred very quickly, and it reduced the number of cases. But now, when they've started these early studies, it looks like herd immunity could be as low as 5 or 6%. It will be interesting. We don't have data yet from the East Coast, but I'm telling you right now, New York is going to have much higher herd immunity than that, and that could be the one point of silver lining to their situation, that, especially in New York itself, is that because they've had such high transmission rates right now, they're going to have high enough herd immunity that if a second wave comes through, that could prevent it from transmitting very well in those communities where they had high transmission initially.
Right. Yeah, that's true. And I want to also just make sure that I go back and say the 1 to 2 year thing is not. I don't suspect that we're going to be on lockdown for that long. I was just saying until the virus goes away completely. Obviously, we know some places are already starting to lift restrictions, as we know. So, I mean, and then there's also been reports too on the West Coast, potentially in California, that maybe they had the virus earlier. I saw that some of those headlines today in the news, and maybe, like you said, you went on lockdown and really tight lockdowns out there early, which I think helped. But the virus was there earlier than suspected. Then possibly the herd immunity could be a little higher as well, right? But then it all just comes back to getting these tests out there so we can actually understand that.
Yeah, and here's an interesting part, though, going back to the story of whether or not we can learn things from the SARS outbreaks back in 2003.
Yeah.
So an example, you don't have to have high levels of herd immunity to stop these viruses. That's what SARS taught us. And here's an example. In Hong Kong, the initial outbreak in Hong Kong had that reproductive number of a 2.9. Meaning that each person at the beginning of their outbreak was infecting almost 3 other people. Right. But even though they didn't have a vaccine and they didn't have a therapeutic, and all they had was a contact tracing, quarantine, and isolation, they were able to knock that reproductive number down to like a 0.4 or a 0.5.
That's awesome.
And then the virus burned itself out. So if we build herd immunity, that's one approach to helping deal with the pandemic, but it's not the only approach. And that's why if we can follow the same successful steps that were made with SARS 17 years ago, we could recapitulate that here and have that sort of success. One of the challenges is that this virus exploded very quickly. And so instead of having one fire to put out through contact tracing and self-quarantine, we have lots of little fires to put out.
Yeah, unfortunately.
Unfortunately. But we're hoping that, through these shelter-in-place methods, a lot of these fires are burning themselves out, because if people are staying at home when they're sick, then they're going to recover and no longer be infectious by the time we open the doors and try and get back to normal activities. Now, because we're still finding cases to varying degrees in different parts of the country, it means we're still going to have a small, as long as you have some cases, you'll have more cases. Right. But if it's a small enough number, then it's containable, because then you can start doing the contact tracing and really focus on those little hot spots. And at that point, it can be contained.
Yeah. And I don't know if you can talk on this or not, but we're using some of these tests that, like these serological tests or antibody tests that you're developing, and finding people that have been exposed to the virus. Are they going to be able to maybe use that data to also, if those people were willing to donate like plasma and serum? Because I remember for like the Ebola outbreak, they were able to use that to help then treat people as well that were sick. Would that also help lead to therapies ultimately in the end as well? Or is that something that, again, just don't know at this point?
Yeah, no, it's actually a very exciting area of research right now. There's been only a handful of clinical studies where they've done, they call it convalescent plasma therapy, but that's exactly the story on that. People who've recovered from the infection have high levels of antibody against the virus, and they're protective levels of antibody. So if we can draw blood samples, collect the plasma from these people who are immune, then we're able to transfer that to the patients who are at most risk of severe disease, or the ones who are on the ventilators that may die if they don't find a therapeutic in time. That antibody has been showing a tremendous clinical benefit for many of these patients. And so there are now currently, there's a nationwide call for people who recovered from COVID-19 to now donate plasma to like a national registry, so that we can we can gather more of this to help people going forward.
Yeah, and even, I mean, I guess if you had it and you were asymptomatic, you wouldn't know to donate, but would those people be eligible as well? If they were able to have access to one of the type of tests that you're developing, could tests see that they had been exposed, and they could donate as well once these become more widely available?
You know, it could be a possibility. Yeah.
Okay. Yeah.
It's interesting. So for most viruses, and COVID-19 probably falls into this category. For most viruses, the more severe your infection, the more the virus is replicated, which gives you that severe disease. And then your body has to produce a lot more immunity to resolve the infection. So people who've have had moderate to severe disease have higher antibodies than people who have mild disease. And then the asymptomatic, there are some people who are showing up with the virus, but having almost no symptoms. They'll actually have lower levels of antibody.
Okay.
But if you can draw enough blood from a person and pool it, you could still find some clinical benefit, because we don't know how much or how little we need in order to get that protective effect in the patient that you transfer it to. But that's another example of where this information could be useful.
Okay, awesome. And let me also just point out real quick too about the whole convalescent serum therapy and everything that we just talked about. I was reading that was originally developed in the late 1890s by a German scientist. I'm going to butcher his name, Emil Behring. Maybe you could say that better than me, but
Okay.
and it was originally a way to cure diphtheria at the time, which I know you're thinking diphtheria for listeners out there. I think my first thing that came to my mind was, I think that's what you would get me when I played the game Oregon Trail back in the day, was diphtheria. But that was at the time the #1 cause of death among children, and he got the Nobel Prize in 1901 for that discovery. So it's pretty cool to read about.
It's amazing. It's incredibly truly old school technology.
Right.
But this was actually they call it, now we have what are called immunotherapies, but before there were antibiotics, this was the only approach you had to treating even some of the bacterial infections, was by transfer of blood from somebody who were able to recover from the disease. And even to this day, when you talk about antivenom, you get a snake bite and you go to the doctor and say, I need antivenom, I got bit by a rattlesnake. That is antibody that they use to protect you with. And same thing with tetanus. If you have a case of tetanus and you're diagnosed with tetanus, you will get antibodies against tetanus to help quench that toxin. So we're already still using this even today, but it's just it's a wild new approach to using it for a virus that did not exist in our consciousness just a few months ago.
Right, which goes into, we'll start wrapping this up, but just into, I mean, we're so thankful that as a country and a world that there's people like you out there, and the physicians and everybody out there that's just come together and are much more knowledgeable about this than obviously 99.9% of the population, and can go from not knowing anything about this virus to knowing a whole lot in just a couple of months and start getting us on track to beating this thing, getting back to our normal lives. And so thank you for taking your time to come on this podcast today and talk to us. And if there's anything else you have that we maybe missed or didn't touch on that you want to share with our listeners, please feel free to throw that out there as well.
I just wanted to thank you for having me on the show. This has been great.
All right. Thanks, Dr. Slifka. We really appreciate it. I just want to do a couple housekeeping things real quick. We're still doing the $100 Amazon gift card giveaway. It's just kind of it's not really on hold. We're just, with everything going on, just things are going to be put on the back burner. We still have it. Keep leaving your reviews and your comments. We might even come up with a new way to give away a couple of those gift cards that might increase participation from the listeners out there and help get more subscribers to the show. The more subscribers we get, the longer we can keep doing this. So share it with your friends and family, everyone you know. We're on Twitter at the LabRat Chat, and our email is labratchat@gmail.com. So, send us a shout out whenever you can. If you have questions, comments about the show specifically, we'll address them as they come in. So thank you again, Dr. Slifka, and thank you everyone out there for listening to the 7th episode of LabRat Chat. Take care.